Welcome, everyone. We just want to start, first of all, by welcoming everyone to the Broad Institute here in Cambridge, Massachusetts. And we've actually prepared extraordinarily good weather for the season for the meeting. Those who trick and treated yesterday had a much better time than in a typical October 31st in this area. We're very excited. This is the next general meeting of the HCA. It actually closes our second year. And Sarah and I, in typical tradition, are going to kick off with a quick overview of what has transpired both since the beginning of HCA and especially for the past 6 months. As you know, we always alternate places, and so Sarah is going to kick us off. I think we're at an interesting inflection point, and that's what we're going to focus on today. And so we're going to talk a little bit about how we're envisioning in our community assembling an actual human cell atlas. Sarah.
Thank you. So welcome, everybody, and happy Halloween. I have my orange scarf, somewhat belated here. We're at a turning point now where we have spent about a year already collecting preliminary data. The first data sets are out. And so what we'd like to have the community start thinking about at this point is not only the data collection, the technologies, the computational approaches, but also how we can coordinate and assemble the human cell atlas going forwards. And as you know, and you've seen a version of this before, where we want to get to is really a periodic table of our cells in the sense of their identity, their location in tissues, and their relationships to each other in state, in development, with an element of disease, with the aim of creating the comprehensive reference map of the types and properties of all human cells. These are the fundamental units of life, the building blocks of our tissues, our organs, our organ systems. And what we hope is that this can then be a foundation for understanding, diagnosing, and monitoring health and disease. You've all seen this, but it's good to remind ourselves of the mission. Again, just to kind of recap our history, many of you are kind of well-established members of the community and have been on this journey from the very beginning. But just to remind you, as Aviv said, we kicked off this crazy endeavor kind of just over 2 years ago. And at our meeting in London in October 2016. And since then, it's been a pretty intense and exciting time with many meetings over the years periodically. And importantly, we've had a few landmarks very recently with the release of the pre-DCP, and there are some nice data sets there, and we encourage everybody to contribute, and we'll have dedicated presentations on that later. And then also I want to emphasize the ethics workshop, where we've started thinking about the access, compliance, new legal developments like GDPR in the EU, and so forth. And so we have also the ethics working group here, and Bartha and colleagues, and Barbara who are, and Rodrigo who have been thinking about this together with others, and also John here who's been spending a lot of time thinking about this. We'll have, going forwards, we're now obviously at the general meeting, going forwards, we'll have the DCP Alpha demo and the interactive registry launch are the things that are happening right now and that are very exciting things we can look forward to. So the community is basically formed by all of us and everyone globally. And what's very exciting is that we, the scientists, have initiated this endeavor, and it's been a grassroots community effort where there's been tremendous commitment sort of from many volunteers, essentially, who are enthusiastic, dedicated, and committed people to get this going. And what's happening now is that the partnership with the funders is becoming stronger, and many funders are committing. We have the Chan Zuckerberg Initiative, and of course you're all, I presume, aware of the Seed Network call that's open now, and that's the deadline, where the deadline is the 13th of November in just a couple of weeks. We also have different initiatives from the NIH, the HubMap, the Brain, and the Cancer Moonshot. The EU and national EU funding agencies like the Swedish and the French are committed to this, and Swedish philanthropic foundations like Wallenberg and Persson. UK funding agencies, Wellcome and MRC has an open call, which is closing on Monday, so that's very exciting, focusing on development and adult match tissues. And then also philanthropic US foundations with a big special thank you to the Kavli Foundation for this meeting support, and the Helmsley and the Manton, who've also been great supporters. So this is you can see a very diverse and in many different ways globally and in terms of foundations, set of organizations that are supporting us. And so what we as scientists need to do is to make sure that these different efforts, these different consortia are integrated across the scientific topics, across the world. And one way of achieving that is through our interactive registry. And just to remind you that this is at humancellatlas.org/joinhca, and incredibly, we now have 1,000 members who are registered. And you can see here how this breaks down across the different countries, hundreds of different institutions all across the globe. And here we have basically the distribution across different areas of research, different organ systems, organs, and tissues. And then also different types of, roughly, categories of scientists. And so this is, I just want to emphasize that we and people here are doing our best to allow this cross-consortium integration to happen via the registry, via the working groups, via the biological networks, and so on. And so this is how we need to work together to get towards assembling the atlas. Also, just for all of you, just to make sure that you're all aware, we've been, thanks to the fantastic team at humancellatlas.org, we've pulled together our first newsletter. You should have all received that if you're in this registry. If you didn't, please check your spam folders. And if you have ideas for items for the next newsletter in a couple of months, then do send ideas in that we can add. So what have we been up to, basically, in terms of coordination and organization? So you may be interested in the publications committee, which is a group of about 6 tremendously dedicated scientists from different countries, and we've got together to start discussing submissions for, let's say, what we consider as an HCA publication. We're actually working as we speak sort of on how to define that, how to assess it, and we've had our first tranche of 4 papers, and there are another 5 that are kind of in the works that have been submitted to publications at humancellatlas.org. So please feel free to send in things that we can discuss. The other work in progress is a data release policy, and this is very important in terms of data submission to the DCP, but also compliance, as I mentioned before, with national and international, local, national, international ethics laws and regulations. And the other element that we've been working on, and this is Charlene and Jay and so on, is HCA identity and how do we maintain consistency and cohesion with things like online tools, logos, and so on and so forth. I also just want to have a shout out for the working groups. So you'll hear presentations from all of these later, but we've got really amazing an analysis working group that got together and just steamed on from the get-go. The standards and technology working groups that you'll hear is really getting going now and is obviously very important for the cohesion across the consortium in terms of technologies and standards, and then ethics and DCP governance and guidance. So that really brings me, I mentioned standards and technologies, and that really brings me to the technology side of Human Cell Atlas, which is the absolute foundation and really the catalyst for why we're here and what makes it possible to build a Human Cell Atlas. And besides the single-cell genomics, I want to emphasize that the spatial methods are just exploding and developing apace. And this is incredibly exciting, both at the RNA level and the protein level, where we've got methods at in terms of different plexities, different sizes of fields of view, different levels of resolution, and they're summarized here, and there'll be more details later from the Standards and Technologies Working Group. And there's a CZI-funded consortium, the SpaceTX team, that's comparing, it's offering some comparisons, some benchmarking, and deeper understanding of how these technologies complement each other. And you can see that here, the group of people who are moving forwards on this. And I just want to give a little shout out to Xiao Wei and congratulate her on the fantastic recognition that she's had in terms of the 2019 Breakthrough Prize. And I want to give her a little clap here. Everybody join me in congratulating Xiao Wei. So the other technology that's obviously been foundational and catalytic for Human Cell Atlas is single-cell techniques, single-cell genomics, single-cell RNA sequencing. And we've got benchmarking, community benchmarking efforts in progress, coordinated by Holger, who unfortunately has missed a connecting flight and will join us later, and Joshua Levin and colleagues. And so these are, again, these sorts of benchmarking efforts are really foundational for the consortium. So just finishing off here before I hand over, I want to say that we're also, as a community, learning a lot of lessons in terms of pipelines and standards. And SOPs are shared on protocols.io. And there are some, very, in a way, kind of mundane, but absolutely crucial lessons in terms of the process of tissue handling, tissue preservation, the process of tissue acquisition. We have the Cambridge Biorepository for Translational Medicine that has this very sort of strictly controlled process for acquiring tissue from deceased transplant donors. This is the clinical pipeline here at Broad with tissue preservation pipelines that are very strictly controlled. And then, of course, these go downstream into tissue processing pipelines. And so these SOPs are actually quite mundane but important. And at this point, I think, do I hand over? Yeah, on you go, Aviv.
Yeah, I'll actually make one more point. How does this go back? Yeah, I'll make one more point here as we transition, that if you read really these little fonts, these pipelines are being built now in order to distribute out to the world. And so, I don't know if this is on. I think now it's on. Yeah. These pipelines are being built in order to spread out to the world. And so, as Sarah said earlier, one of the key goals is to make things in the consortium work broadly and in an integrated way. It's going to be an ongoing challenge for the next year or two to get all of that spread, coordinated, synchronized, cohesive. And it starts with mundane things, as Sarah said. Sometimes they sound to people like, it's not the next paper that they're going to write, but they lead to processes that are robust and that allow an entire community to analyze a shared set of samples. By the same token, they lead to processes that allow us to process a very broad range of human tissues. And so not just the first tissue that succumbed to a technique, but really bringing every technique to work across the human body. And as Sarah already alluded to, the ability to share these protocols and activities across the consortium members in an open way. And in this, we're extremely grateful for the partnership with Protocols.io. And I also want to give a shout out to CZI, who has really spearheaded the requirement of grantees to share lab protocols, not just code and not just data, but also methods that they use in the context of Protocols.io. So far, there are 58 public protocols, meaning everyone in the HCA community can actually view them, each of these 167 members. People also develop their protocols first in private mode until they kind of feel they have something that they actually believe works. And that allows them to then quickly share it across the community. People write questions to each other and comments, and they suggest improvements. And it really leads to a much faster advance, similar to what has happened in the open source code community. One of the particularly exciting moments for us is the fact that the DCP is going to demo to you today their first end-to-end offering that starts with 2 types of data sets from single-cell RNA-seq with a SMART-seq2 method and data coming from droplets, and both provides access to the raw data from these methods and to pre-process data that could then empower the scientific community. It includes appropriate documentation. They have a set of lined up beta testers for this phase and the ability to obtain the expression data and then proceed with it downstream. And again, this emphasizes to us the fact that both data will be fed through this and be obtained through this, but the community needs to come together to define what they want more. What other questions, what other challenges, what other data types are going to come, what additional analytical efforts, and this entire process, while performed by the engineering teams, the 4 engineering teams that build the DCP, involved critical contribution from the analysis working group in really guiding the process of knowing how you move from raw to pre-processed data, and we're extremely grateful for their contributions. And so in the tradition that we actually started in Hingston, we're always going to give a little bit of snippets into what is actually being collected. And this was really the year that marked for us real data generation in the Atlas. We'll have lightning talks to drill a little bit more into the biology of these. So this is really mostly for just the pleasure of seeing the data streaming in. And I'm going to start with the kidney, because 6 months ago, we didn't have a kidney slide. There were groups here and there. Now there's really a community coming together, putting together projects, coordinating across countries, and in the process covering both the developing and the pediatric and the adult kidney. In fact, the first paper on that is already out. More digging deeply into the developmental atlas, and then working on adult kidney tissue in both health and in disease, and obtaining samples from both living donors and deceased organ donors. A similar expansion, a dramatic increase, has happened in the context of the skin cell atlas. There is now work, again, across multiple teams and multiple countries, and here you can see also the integration between single cell measurements and spatial measurements, and between very broad approaches that look at large numbers of cells and deeper approaches that look at those cells very carefully in specific contexts. And just by getting an idea of the many different names on every slide, you can see the scale of the activity. The lung, as we know, has already delivered at least several key discoveries of things we didn't know were there in the lung and the airways. And again, across the community, we're taking several different strategies as to how to build an atlas. From looking at a larger number of donors but fewer cells per donor to get a real assessment of what is going on in the context of variation and obtaining samples from living individuals, to looking at a very large number of cells from a smaller number of donors in order to know that you reach completeness, especially around rare cells, and to have complete ability to sample an entire lung by working with organ donors. And by having both efforts and coordinating between them, we know that we can get a complete view of the cells. And then in parallel, expanding to additional types of data, especially around single cell epigenomics, and starting to look at processes such as lung differentiation or at different anatomical locations. Similarly, in the gut is very long and made of many parts, but there's now work in the colon, in both health and in different types of diseases, and in the ileum, and in the esophagus, that is going to allow us to sample across this very complex organ using different types of methods, and really also an effort to get to all the cell types in the gut, including challenging ones like the neurons. There's been a lot of effort around developmental cell atlases, the placenta, processes like blood development in the fetal liver, the heart, and there are additional tissues. You'll hear more about the brain in a dedicated session today. This was really in order to both make people happy and make people worried. So making you happy is easy, right? Here's data, and we're making discoveries, and it's all wonderful. Making people worried is sometimes a little bit harder, but I think this is actually a critical inflection point for HCA. And it's one around which this specific meeting is organized. We know now that we can actually collect the data. We can collect it robustly and reliably. We can collect it from living people. We can collect it from organ donors. We can collect it from postmortem. It is absolutely abundantly clear that one can profile single cells and one can analyze single nuclei. It is also now increasingly clear, and I think Sarah really showed that beautifully, that there will be the spatial techniques there to also get that. That will get us a lot of data. It will also get scientific insights and papers and discoveries.
But what we are congregated around is not just doing our own individual research and specific research projects, but it's really to build an atlas of the human cells. And that's a higher order problem. And so it's not just about obtaining individuals and breaking it down to the pieces and measuring those. It's also about taking all of these pieces and building them back up to something that we can say is the actual atlas. At first incomplete and in draft form, and then more and more complete. And that's a tough problem. And it's a tough problem whose time has come. 2 years ago, there wasn't this. And so we had to figure out how to get there. We now know we're there. We have the connectivity, we have the community, we have the technical ability, we have the demonstration. I think we've even convinced people that it actually matters and it is going to serve us extremely well to do it. But we still have to actually do it. And what we will discuss in this meeting, especially in the breakout sessions, and as we report them back, is specifically how. So here's a cartoon view of some of the aspects we have to worry about. We have to collect multiple samples because there are multiple humans. We have to find ways of placing the right landmarks based on prior knowledge and newly acquired ones in order to know that it's the same thing. We need to find ways of aligning those to each other, to find what are the key features in them, what are the cells that are shared between them, the histological structures, the anatomical structure. And this is not something that is done as a one-off. It's a gradual, iterative process, and a critical component of it is reaching an annotation that we all agree upon. The analysis working group is going to talk about that a lot more very shortly. And then we need to place it in a reference coordinate system that somehow remarkably works both to organs that have these complex stereotypical structures like kidneys or lungs, systems that span the entire body like the immune system or the vasculature or the skin or fat deposits, and these more dynamic and loose structures that form within the context of the tissue. There's plenty of technical and biological and computational problems along this. At the same time, this is something that calls for cross-consortium coordination of our efforts and for monitoring of our progress, for knowing where we actually stand. And that's something that both the organizing committee, the working groups, and every participant in the consortium has to be worried about and working on. We are worried about it, and we are working on it, and we want everyone to think about this very carefully over the next 2 days. How do we assemble, not just how do we collect? We know how to collect now. It's been demonstrated successfully. We'll continue doing this. We have a roadmap on how. We want to also assemble it together and make sure that we have a stellar and beautiful atlas at the end. And so with this, we will end this part of this morning's presentation. This was actually made by colleagues in the UK. It turned out really cute. I guess a meeting around Halloween has its own special features, and wishing you a very happy Halloween.